Hello everyone, today we are going to see about a food microbiology. A food microbiology is a branch of microbiology. They focus on the study of microorganisms present in the food and their impact on the food safety, preservation and the quality. It examines various aspects such as a microbial growth, spoilage, fermentation and foodborne illness. Next we can see about the significance of food microbiology. Food microbiology plays an important role as well as a beneficial and a harmful. First, we can see about ensuring food safety. The most of the microorganism causes food spoilage and a contamination. It can lead to a ensuring a food safety. A microorganisms such as bacteria, virus, molds, and yeast can contaminate food products at a various stage of an distribution. Some of these microorganisms can cause a food illness, posing a serious risk to public health. A food microbiology plays an important role to help the identifying and preventing measures and the stringent quality protocols. Next, we can see about maintaining product quality. Microbial contamination can also lead to a spoilage of food products, and these are result in the changing in the taste, texture, odor, color, microbial population, and the implicating appreciated control measures. Food microbiology helps in the preservation of quality and the shelf life of a food product satisfaction and the brand reputation. Next, we can see about a compliance and with the regulatory standards. Regulatory agents has been worldwide has established a triggered standard and the regulation to ensure the safety and the quality of the food product. These regulations often include a microbiological criteria for various food categories. Next, we can see about preventing a foodborne outbreaks. How can we prevent a foodborne outbreaks? Microbial contamination in the food products can lead to a foodborne disease outbreaks, which can have a several consequences for a public health and a food industry. Food microbiology helps in the earlier detection of microbial hazards and enabling a timing intervention to prevent a outbreak. By identifying a source of contamination and then implicating corrective action, food manufacturers can effectively mitigate a risk of foodborne illness. Next, we can see about supporting process control and optimization. Microbiological analysis also provide a valuable insights into microbial dynamics within the food processing environment, including raw materials, equipments, and personnel. By monitoring a microbial population and trends, food manufacturers can identify a potential source of contamination and implement preventing measures to maintain the hygienic conditions and optimizing production process. Next, facilitating product development and innovation. Food microbiology research contributes to develop of an innovation technologies and approaches for improving a food safety and quality. These includes the development of rapid detection methods, involve a preservation technique, probiotic formulation, among others. By leveraging advancement in the food microbiology, the food industry can continuously innovate and meet evolving consumers' demand. Next, we can see about a microbial hazards. The microbial hazards in the food product pose a significant risk to the public health, can lead to a foodborne illness. These hazards can originated from a various sources, including raw materials, processing environments, equipments, personnel. Some of a common microbial hazards in the food products include. First, we can see about a bacteria. Bacteria is present everywhere. Some of a bacteria can be a beneficial and some of they can be harmful to a public health. First, we can see about a salmonella. Salmonella is found in the food raw material, poultry, egg, and unpasteurized dairy product. Can see a salmonellosis with a symptoms such as a diarrhea, fever, and abdominal cramps. Next, E. coli. Certain strains such as E. coli 0175 and 7 or associated with the contamination from uncooked ground beef, raw vegetables, and unpasteurized milk can cause several illness, including a bloody diarrhea and a kidney failure. Etc. Next, we can see about a virus. Norovirus, often associated with the contaminated water, raw produced and selfless, can cause aquatic gastrointestinal with the symptoms such as vomiting, diarrhea, nausea, and stomach cramps. Hepatitis A virus can be transmitted through the contaminated food and water, causes hepatitis A, characterized by symptoms such as fever, flagellated, nausea, abdominal pain, and jaundice. Next, we can see about molds. Certain molds can produce a mycotoxins such as an aflatoxins and orchotoxins. Orchotoxin A, which contaminates the grains, nuts, and dry fruits. Prolonged exposure to the mycotoxins can lead to the various health problems, including liver damage and cancer. Next, yeast. The yeast can be used in the several cooking product for a fermentation process. It causes some illness to public health. While most yeast are not harmful, certain species can cause a spoilage of food products, especially those with a sugar or acid content. Yeast contamination can result in the fermentation of flavors and texture changes in food. Parasites such as Giardia or Toxoplasma gonorrhea can contaminate a food and water, leading to a parasitic infection with the symptoms of ranging from mid-gastrointestinal disinfection to several illnesses. Toxogenic bacteria. Some bacteria can produce a toxic substance that it become a toxic. Certain bacteria produce a toxins that can contaminate a food and cause a illness even if the bacteria themselves are not present. Example, Staphylococcus aureus, which produce a heat-stable enterotoxin in food. Uh, Next, we can see about a microbial analysis. How can we identify the microbial contamination in the food? We can see about a microbial analysis. Microbial analysis involves various methods of detecting, enumerating, and characterizing the microorganism present in the food. And the environmental samples. There are several methods. First, we can see about a culture-based methods. In culture-based methods, we can see about three methods: plate count agar, selective and differentiation media, enriched culture. While using a plate count agar, while used enumerating total viable bacteria in food samples, a nutrient agar are inoculated with a sample and incubated, and colonies that develop or counter. Selective and differentiation media such as a McConkey agar, Elixin methane blue agar, and Salmonella flagella agar are used to selectively isolate specific groups of bacteria based on their biochemical characters. Enriched culture samples are incubated in the selective broth media to encourage a growth of a target microorganism before plating on the agar plates for enumeration. Next, we can see about molecular methods. In molecular methods, food detection can be done with three types: polymerase chain reaction, loop mediated isothermal amplification, next generation sequence. And it becomes an immunological. Next, we can see about immunological methods. In immunological methods, we can detect and enumerate a food microbial contaminant through the ELISA linked immunoassays, that's ELISA, and immunomagnetic separation. Next, biochemical and physiological test, API strips and metabolic activity test, flow cytometry, fluorescent activated cell sourcing, microscopical examination, direct microscopical, sensor analysis, organophonetic evaluation. And what are the challenges if the food become contaminated, and how it can be reacted within the body? We can see about the challenges. The food challenges become a complexity of a microbial communities, detection of a low values of contamination, a rapid detection and response, valuation and verification methods, cost and resources, regulatory complaints, emergency microbial threats, supply chain management, etc. From these we can be from this the food can be challenged to be detect and enumerate microbial contamination where it is found in the food. And next, we can become a conclusion. In conclusion, a quality control in the food microbiology is essential for protecting public health by preventing a foodborne illness, maintaining a food product quality, complying with the regulation, and safeguarding branded reputation. However, the field is not without its challenges, including a complexity of a microbial communities, a need for a rapid and sensitive detection method, a resource constraints, regulatory complaints, and emerging microbial threats. Thank you.